and microfluidic platforms to understand structure at the molecular level, as well as trying to look at whole cell stiffness at the whole cell level for a variety of different problems. So today I'm going to stick to the looking at the molecular structure of DNA and DNA protein complexes, and more specifically looking at the nucleosome array system. The whole day we have been talking about why this is very important, so I'm not going to go into that. All of us are interested in looking at how do these nucleosome structures are formed, how do they move around, how do they get broken apart, or how do different chromatin remodeling enzymes change the structure of these nucleosome arrays. So what we do in our lab is to use something called as a nanopore platform. Those who don't know, it's basically a very thin membrane which has a very tiny hole, and we basically have salt solution on both sides, and we apply a potential which allows for a nanopore current. So that current goes through this particular pore. And whenever there is a DNA that is going through this particular pore, you are going to have a signature. So this is the open pore baseline, and whenever a DNA goes through it, you basically see a specific electrical signature for a DNA that is translocating from one side of the nanopore to the other side of the nanopore. It's like one molecule electrophoresis gel. But we are very sensitive that if your molecule has a specific structure, double the molecule, we have signatures which sort of correspond to that. If you have some sort of a substructure, we will have signature which basically corresponds to 2 DNA strand and 1 DNA strand. And similarly, we can basically have a very nice distinction between 1, 2, 3, 4, 5 DNA strands. The idea is just to say that we have a very good resolution, that whenever you have a particular molecule crossing your nanopore, depending on the structure, we'll have an electrical signature that corresponds to that structure. There are 2 points which will come sort of very important later, that the number of levels correspond to the number of structures that are there in any given polymer, and the depth of these levels corresponds to the size of the structures. So we'll talk about number of structures and the size of the structures as we pass a nucleosome array through a nanopore. All right. So in my lab, we make these nanopores using glass capillaries. Forget about how we make it, but we can basically make it in a way that we get very similar signatures, that using glass nanopores, which are conical in nature, whenever DNA goes through, we basically, based on the structure of the molecule, we can get electrical signature that tells you about the structure of whatever is going through the pore. Very quickly, the first application was for just detecting different folds of the DNA, as well as looking at comparison of regular nucleosome versus Senpei nucleosomes. And the idea was to see if the octosomal state of the regular nucleosome remains same for a Senpei nucleosome. This is slightly older work, but this is pure detection through the nanopore. Since we have an asymmetric nanopore, we are also trying to see that what happens to the polymer dynamics when it is going from outside to inside versus from inside to outside. We see some very interesting features, and we are currently sort of analyzing all these data. We have looked at voltage dependence, size dependence, and all those things. Then comes looking at the structures which are sitting on a given piece of DNA. So here we are looking at supercoiled DNA, and depending on the supercoiled branch, the size of the supercoiled branch, we get very unique signatures that can quantify this branch size of any given supercoiled DNA. Not only that, we can use an enzyme to nick this supercoiled DNA, so that the supercoiled nature, so this is as a function of time, the supercoiled signature dies down and a linear DNA basically rises up. So you could also look at enzymatic reactions using this particular system. Of course, you can't go less than 5-minute measurements, but with the time scale of, let's say, 5 minutes, one can basically look at how does any given enzyme change the structure of the DNA molecule inside your test tube. The last piece here is that if you have a protein that is sitting on a given piece of DNA, we have the DNA level, and then we have a spike that corresponds to the location of the protein that is sitting on that given piece of DNA. You switch the location of the protein, you basically also switch the location of the spike. You switch the number of protein which are sitting on the DNA, you basically also see that in the number of spikes that basically show up in your data. But all of this is to simply say that what happens now that if we look at a nucleosome array that is basically thrown through this nanopore system, And the idea is that can the electrical signal that we are getting, that we are going to get from these nucleosome arrays, can we say something about the different kinds of structures that a nucleosome array essentially forms? So in this case, what we saw was, so this is the 5 kb DNA on which we have formed our nucleosome array. We have an asymmetric array so that almost all the nucleosomes sit on one side because this is a repeat of the 601 sites, and the back side of the DNA is just bare DNA. When we have no nucleosome sitting, we basically get the background signature of 5 kb DNA in the current experimental conditions. But when we start to look at the nucleosome arrays, what we start to see are these very complex things which basically correspond that, so this individual event is one molecule of the nucleosome array that is going through the nanopore. And what we notice is that it basically has a very large number of substructures, sub-levels. So you have different levels, and each level has a specific depth, which has specific size. So to cut the long story short, yes, we have a library of different kinds of complex events. Almost all of them are multi-level events. And what we feel is that you have multiple nucleosomes which are within the sensing range of the nanopore, that is basically giving you these multiple levels. But we started to quantify what is the size of these levels. And we started to see something very interesting, is that if you start measuring these multi-level events of every array going through the pore, as you start to increase the voltage, the number of multi-levels start to decrease, as well as the depth of these levels start to decrease. So the size of the structure is reducing, as well as the complexity of the structure starts to reduce. So this is the first time that we have seen that just by increasing the voltage. So this is the voltage which is applied across the nanopore and your so if I draw it very quickly. So this is your nanopore, and this is the structure that is basically going through, and you are applying a voltage across the system. All right. By applying higher and higher voltages, what we started to see is that the complexity of these structures started to vanish and their sizes started to decrease. So first thing that we thought was that probably we are spoiling our sample. Something is going wrong with our nucleosome arrays. So we started to check. So this is the quantification of these how many levels are there. But to make sure that our sample is not going bad, what we basically checked that we can work at low voltage, then high voltage, back to low voltage, back to high voltage. Every time we are measuring at low voltage, we see these multi-level events, lots and lots of structures on every nucleosome array. And whenever we go to high voltage, we basically lose all that multi-levelarity, that complexity of the event. If we can also randomize the sequence in which we are applying the voltage, we again see the same thing. We can choose any sequence. Every time we are at lower voltages, we are seeing these complex events, and at higher voltages, we are not seeing these complex events. So what we thought is that one of the possibility is that we are essentially breaking these nucleosomal structures, these octosomal nucleosomes, into smaller structures. To verify that, I'm leaving out a lot of control experiments for the sake of the time. So this is a mononucleosome, which is an octosome, which is a mixture of an octosome and a hexosome. And that gives, in the delta G histograms, these two peaks. And we'll notice that as you increase the voltage, the octosomal population goes down and the tetrasomal population basically starts to come up. So what we are doing is that we start working with the complete octosomal nucleosome, and with the application of higher voltages, what you are doing is that you are essentially rupturing these mononucleosomes into hexasomal or tetrasomal nucleosomes. So from here, we can basically get the exact delta G values for these 2 or 3 different types of structural states, and we can sort of connect those numbers in these delta G histograms, that in the sensing range of our nanopore, we can get 3 nucleosomes at a time. So we should have levels corresponding to a trinucleosome, dinucleosome, mononucleosome, hexosome, and tetrasome, which should be these dotted vertical lines. But we also have these mixed structures, which is the full nucleosome being broken down into these hexosomes and these tetrasomes. So what we have essentially shown in this particular project is that at low voltage, you might have the complete nucleosome, but as you start to increase voltage, you start to break this octosomal system. And what we feel is that your DNA is essentially a negatively charged, you basically apply a minus and a plus. Your DNA experiences a force in this direction because it is negatively charged. your your histone octosome essentially is a positively charged protein which is sitting on the DNA. So it gets a force in the opposite direction. So that we believe is the best explanation, where we basically start to see that the octosome starts to break into these hexosomes and these tetrasomes. So this, what we call is the first demonstration that this is a voltage-dependent rupture of the nucleosomes. I think that this is a very good system to start and understand what are the stability features of any nucleosome or its modified nucleosome or some sort of an epigenetically modified nucleosome. How strongly does it bind to a given piece of DNA, and its sequence specificity can, all of these things can be studied by applying a direct electrical force to the system. This is slightly different from a typical optical tweezer or an AFM-based measurement, because there you are applying force directly to the DNA, which is here, and you're hoping for the protein to pop off. In this particular case, you are applying force directly to the protein-DNA complex in an opposite way so that you can break the system. Thankfully, that this particular protein is an octosomal, so it breaks down into smaller pieces. If this was a single protein, we think that the whole protein will just pop off in one shot. So we are doing further experiments with Cas9 essentially to basically see if we can pop off the entire protein just by applying voltages. Just the last slide, that what we also want to study is that using an optical tweezer, we basically want to hold any given nucleosome in this nanopore region and then start to increase the voltage so that one can dynamically see this rupturing of the full nucleosome into its smaller pieces. So if that can be done, that will be interesting. If that can be done, then one can also look at some sort of an nucleosome, sort of condensation because of the internucleosomal interaction, or using these architectural chromatin protein, which can form local condensates. So these are the kind of problems that we are essentially looking at. So thank you, everybody. Most of the work that I've explained is of this student Sumant, with a little bit of Sukanya and Puneet's work thrown in as well. Thank you very much.
Thank you, Gautam. Questions? Yes.
Nice talk. So I have a naive question. Like at a given voltage, when you apply, why do you see a mixture of nucleosome? Suppose you are increasing the voltage, keep on increasing, and you are seeing a shift towards tetrasome, the hexasome. Why not a single peak at hexasome or a single peak at tetrasome?
Because when the octosomal nucleosome breaks, it can only break into its stable substructures. So if heptasome is not a stable structure, it will not form. So it might form a hexasome, which is reasonably stable, and then the tetrasome. So again, our best guesses there is that the two populations that we see are the hexasome and the tetrasome, which is known in literature that they have a reasonably stable system.
I'm asking, at a given voltage, why are you seeing this mixed population, not a single population such as hexasome only or a tetrasome only?
Oh, so that is the heterogeneity in the nucleosome array that we are preparing. Anytime when we prepare a nucleosome array, the very nature of this preparation is that you will get heterogeneity. So in general, people who make the nucleosome arrays, if you see something sitting on the DNA, you hope that all of them are complete octosomes, which is not entirely true. So for example, even when we make single mononucleosomes, I showed that it is sometimes a mixture of the octosome and the hexasome, right? So one has to be very careful as to what is the stoichiometry of each ball that is sitting on the given piece of DNA. So it is hard to sort of manage that.
Hello. Yeah. So my question is regarding how does the conformation changes if you tune the depth of your pores. Basically, I'm interested in whether the polymeric degree of freedom is playing a role or not.
You mean depth meaning this length?
Yeah.
We can. It is something that needs to be measured. The only thing what happens is that the size, or the length of the pore basically decides how many nucleosomes will be averaged out in a given level.
Yeah.
Right? So ideally, it should be as thin as possible so that you can see. Ideally, when I showed you the single protein data, we were hoping that we're going to get the DNA and then we're going to get nucleosomes sitting like this. Okay, if there were 4 nucleosomes. But what we end up seeing is these multi-level events, because all 3 of them are within the sensing range, and the level that you are getting is the sum of all the 3. So you get this averaging effect, which is pretty typical for the nanopore community. So we try to make this as small as possible. But if you make it long, you'll get more averaging. That's
no. I think if we make it long, then essentially the entropic degree of freedom of polymer, which would force him to crumple, that should also play a role. So in the other limit may not, this quanta may not be realizable. That's what I'm think.
Explain me later on the board.
Yeah,
I'll
sure.
be able to tell more.
Sure, thank you.
Hi, very interesting talk. So I wasn't aware of glass nanopore sequencing for DNA. So usually the commercial ones use protein nanopores, I think.
Yeah.
So just getting back to plain DNA sequencing, you said it's very accurate. What's the error rate, and is this, because for the commercial nanopores, it's still something like 1% error rate. It's much, much higher than Illumina.
Yeah,
So-
glass nanopores are no better than the protein nanopores.
Okay.
It is either same or worse.
Right.
Yeah, so that's why we don't, I don't work in sequencing.
Okay.
I've done that for decades, but then now I just only try to use the same idea, but to look at structural features
Yeah, that's
on
very
some interesting
yeah.
polymers rather than the sequence.
Okay, thank you.
Yeah. So this is in a way sort of an epigenetic sequencing. The whole aim later is to basically see if one can read off the epigenetic state based on either the size or the rupture forces. So that is something where we are trying to head.
It sounds exciting. Thanks.
Yeah, Gautam.
So if I understood well what you just said, so basically the complexities that you have at low voltage is just due to the fact that you have several histones that are on the same. So,
But we thought it should be an integral multiple of one ball, 2 ball, 3 ball. But what we started to see were sub-integral multiple, and that's when we saw that I think that we are breaking these things. And that's when we measure with higher voltages, we sort of confirmed that yes, we are breaking these things.
and can you play with the salt concentration or with different ions? So does it change a little bit the picture, or
No.
regarding especially the?
So we have tried sodium, lithium, potassium, and different voltages, different salt. So usually every DNA protein complex has a happy place. So for this guy, it's 0.5 molar salt and lithium chloride. You get beautiful clean data. If you try with, let's say, sodium chloride, you get a lot more sticking between the sample and the glass surface. So your pore blocks a lot. So it takes quite a bit of time to basically find the best spot or best conditions for these experiments to work. So yeah, if you change the salt, we have more messy data, but not different data.
But does it have an impact on basically on the last point where you were looking at the different composition of in hexasome and?
Nope.
No. Okay.
Yeah, Gautam, very nice talk. So I have one question regarding the heterogeneity. So if you use something like a Widom 601, which is strongly positioning.
This is what it is.
Oh, you use Widom.
Yeah.
Okay, then okay. So the other question.
So we intentionally made a sub-saturated because we wanted to see some sort of heterogeneity.
I see.
But
Okay.
we thought it should be a multiple of the whole ball.
Right.
But the broken ball was the interesting part.
Right. So the other question is, will you be able to like compute some free energy cost of forming these, subnucleosomal particles and compared it with what you get from optical tweezer experiments, like
Yes.
Bustamante
Yes.
and others?
So we are thinking in that direction. So this is something that we finished just last year, but we are moving in that direction too. Yeah. I'll be happy to talk to anybody who wants to do those calculations.
Thank you.
Okay, if there are no more questions, let's thank Gautam and all.